In this fourth lesson of Chapter 7, we're going to be looking at other types of respiration besides aerobic cellular respiration, where there would not be oxygen as the final electron acceptor at the end of the electron transport chain. And this is something that happens in your own body. If ever you're in a state of anaerobic respiration, when your body is using oxygen faster than you're bringing it in, or you're working faster than you can breathe. So without oxygen to pull the electrons down the electron transport chain, oxidative phosphorylation doesn't happen, nor does citric acid cycle or pyruvate oxidation. So in fact, the only payout of ATP here would be the two from glycolysis. Now, in this scenario, this is anaerobic respiration. No, I'm sorry, this is fermentation because it does not use the electron transport chain. I have this on the next slide. Fermentation does not use the electron transport chain. However, anaerobic respiration does still use the electron transport chain. So you might be wondering, well, how can the electron transport chain operate without oxygen? Well, there would have to be another compound that, like oxygen, is electronegative and is going to pull the electrons towards it. So here is one example of a different compound besides molecular oxygen, which would be the final electron acceptor, and that's sulfate, or SO4 2-. So the nature of sulfate, it does have oxygen, but the compound itself is still electronegative and will draw those electrons towards it. Similar to nitrate and carbonate. These are also alternatives to oxygen as the final electron acceptor. So with this, there would still be up to 32 ATP produced from, this would be anaerobic, not aerobic, found a typo here, respiration. Again, the criteria for anaerobic respiration has to do with a substance, a molecule that's similar to oxygen in that it is electronegative. It's going to drive that process of electrons towards it. So remember the trend for electronegativity, where fluorine is the most electronegative, followed by oxygen, nitrogen, chlorine, bromine, and so forth. So where would something like this happen in the living world? Well, these very extreme places are examples where anaerobic respiration takes place, and this would be done by prokaryotes in hypersaline lakes, hot springs that are really acidic and hot, anoxic muds, which have no oxygen, hydrothermal vents at the bottom of the sea where light doesn't penetrate the ocean. So these are the extremophile prokaryotes that you might remember we talked about in one of the first lessons of the class. Where these are in the domain Archaea, or they're known as the Archaeobacteria, and they are prokaryotic, so they don't have nuclei in their cells, but they can still undergo respiration in order to make ATP. However, they do it without oxygen. So let me go back to the diagram here. So if this would still happen, the process of cellular respiration for anaerobic respiration would still result in the same outputs of ATP. The difference here is, again, that electron acceptor molecule is not oxygen, it's something else. It's like oxygen in that it can draw those electrons towards it. But if there were no substitute molecule for oxygen, then this would be the case here, where all of the processes after glycolysis would not occur, and that would be fermentation. There are two types of fermentation: lactic acid fermentation and alcohol fermentation. Now, at a quick glance, the diagrams on the right look pretty similar, but they are different in some ways. So there are similarities and differences between the two types of fermentation. Both of them start with glucose. Both of them still require NAD to be the agent that accepts the electrons from glucose. So notice we have NAD is gaining electrons from glucose, becoming NADH, and then the NADH carrying electrons is going to reduce pyruvate, and in lactic acid fermentation on the left, the product will be lactate molecules, and in alcohol fermentation on the right, the product will be ethanol and CO2. Let's take a closer look at these types of fermentation. So step by step here. In alcohol fermentation, the glucose undergoes glycolysis. You still get the 2-ATP and the 2-NADH. And then it results in the 2-pyruvate. Now, this is where it differs from cellular respiration with oxygen, or anaerobic respiration without oxygen, but with a similar electronegative compound that still causes an electron transport chain to occur. So here, the pyruvate gets reduced with the electrons from NADH. 2 CO2 are given off. The resulting compound initially is acid aldehyde, which ultimately, after this whole process is completed, becomes ethanol, 2 molecules of ethanol for every 1 glucose. Now, ethanol is a 2-carbon organic compound, and we started with glucose, which is 6, which split, became 2-pyruvate, so there were still 6 carbons, but we end up with only 4 carbons between the 2 ethanols. So where did the other carbons go? Well, they were given off as carbon dioxide. So think about alcohol fermentation. What you might already know about alcohol fermentation from foods that you eat, beverages that you drink, if you're over 21 years old, of course. You get that fizziness usually with the alcohol. That's the CO2 given off. And then the ethanol itself is the alcohol that would be consumed. Bakers that work with baker's yeast, the yeast themselves are undergoing alcohol fermentation. The CO2 is what causes the dough to rise, and then the alcohol, sometimes it smells like alcohol when you're baking or making bread, and that's the ethanol, and it's burning off, so you don't end up having the alcohol content in the bread. Okay, so this is alcohol fermentation. Similar to that, lactic acid fermentation starts with glucose, which undergoes glycolysis, resulting in 2 ATP, 2 NADH, and 2 pyruvate. But the pyruvate, which again is a 3-carbon compound, so you have the 6 carbons here retained from the original glucose, and then the resulting compound that's reduced by the electrons from NADH is going to be lactate. Lactate is a 3-carbon organic compound. So there's going to be 2 of them for every glucose. So all 6 carbon are retained in the organic molecule. There's no CO2 given off in lactic acid fermentation. So let's look at some examples of where these processes occur in nature. Alcohol fermentation is naturally done by yeasts, type of single-celled fungi, and some bacteria. Of course, humans have learned how to harness the capabilities of the yeast and the bacteria for their own benefit in brewing alcoholic beverages and in baking, as we discussed already. Lactic acid fermentation is also done naturally by yeasts, type of fungi, and other bacteria. But humans, again, have learned how to utilize this to their benefit for making cheese and yogurt. Aside from the food and beverage applications of fermentation, fermentation also happens in our own body. This is the case I was leading off the lesson with, when if your body is working very hard and you're your body is using oxygen faster than you can give it more oxygen. So then it goes into a state of lactic acid fermentation. So what's actually happening during lactic acid fermentation is that you might start to feel like burning in your muscles. Now, part of that could be just the tears of your fine muscle as you're doing an intense workout, like weightlifting, for example, or running for a very long time. If you're not trained for that, you're causing fine muscle tears. But it could also be the buildup of lactate through lactic acid fermentation. Glucose is going to be converted to the lactate. Again, there's no oxygen present. um, Lactate will quickly be absorbed by the bloodstream, where it's then taken to the liver for processing. It can be converted back to glucose, which then can be reabsorbed by the blood and then used as a source of energy, or the glucose can be stored as glycogen in the liver. So there are different fates of this process, but the point being that this is a natural mechanism that the human body has for continuing to be able to do work. Remember, a little bit of ATP is going to be made here um, without providing it with an electron acceptor molecule. So with that of discussion of fermentation and anaerobic respiration, I want to go back to something I mentioned in the beginning of Chapter 7's lessons, that we were modeling this process using glucose as the substrate for cellular respiration and fermentation. But this is not the only substrate that can be utilized by an organism to make ATP. In fact, proteins, fats, and carbohydrates, such as glucose, are all potentially useful for providing a source of fuel for either going directly into glycolysis or even later stages, as that's where all these arrows are coming in at later stages. The whole point, though, is to be able to convert that food, that potential energy, into a source of energy that the cell can use to do work. So just have an understanding it's not just about glucose, even though we've been spending so much time talking about glucose. In addition, glycolysis in the citric acid cycle can also provide building blocks for other anabolic pathways. Anabolic pathways, remember, are pathways that build more complex, larger molecules from smaller molecules. So taking some of those derivative products from out of glycolysis, out of the citric acid cycle after the glucose is oxidized, those can come essentially out of the process of aerobic respiration and be used to synthesize other compounds. So that's what's being shown here. You can see, for example, pyruvate could go into the citric acid cycle as acetyl coenzyme A, or to this other compound called oxaloacetate, and ultimately derived be used to derive other amino acids like pyrimidines and purines. So just again, have a basic understanding that it's not always just a straight shot from glucose to aerobic cellular respiration resulting in ATP. There could be other avenues for that glucose to have a purpose in the cell. And this concludes the lesson for Chapter 7.